Hello there! Today we'll be learning about enzymes, specifically the dehydration synthesis and the hydrolysis, with the help of hydrogen, oxygen, glucose, an allosteric inhibitor, water, a competitive inhibitor, and of course, enzymes. Let's go do some chemistry. Let's see how glucose molecules can bond together through dehydration synthesis. Dehydration synthesis is the chemical process in which a water molecule is removed from the substrates, making them form a new bond. Enzyme helps speed up a process, in this case, dehydration synthesis. The substrates, glucose A and glucose B, fit into a particular enzyme. And an important note is that the shape of the enzyme determines the function. Glucose A and glucose B fit into a particular part of the enzyme called the active site, where they are either bonded together or broken apart. During dehydration synthesis, a water molecule is removed. This makes the two glucose molecules form a new bond through the oxygen atom left over. Glucose plus another glucose makes maltose, which is a type of sugar found in brewed beer. Let's see how glucose molecules separate through the process of hydrolysis. Hydrolysis is the chemical process in which water is added to the substrates, making them break apart from each other. To reiterate, an enzyme helps speed up a process. In this case, hydrolysis. Glucose A and glucose B are bonded together through the oxygen atom. They then travel to an enzyme that has an active site that can fit both molecules. A water molecule is added to the bond, adding 2 hydrogen and 1 oxygen back to the glucose molecule. You're thinking, Annalise, what if the enzyme's damaged? Can the substrate still bond or break? Well, that's a good question, and I have an answer. There are 2 inhibitors. One is a competitive inhibitor. Competitive inhibitors attach themselves to the active site, but don't react. It merely takes up the space of the site, blocking out the substrates. An allosteric inhibitor fits onto the enzyme and reshapes it. This inhibitor is very important due to its ability to manipulate the shape of an enzyme because, to reiterate, the shape of the enzyme determines its function. By changing the shape of the structure, it also changes the active site. If the active site is reshaped, then the substrates can't fit anymore. Important to note that there are also allosteric enhancers. Both allosterics attach to the enzyme at the allosteric site. The enhancer also changes the shape of the enzyme, but it reshapes the enzyme in order to make it work again. That's all the time we have for today, guys. Thanks for watching. Peace.